Okay, so today I'll be talking about hypothesis testing. So now this is part of the class that you've all been waiting for, and we can apply what we know about confidence intervals and the normal distribution in order to test hypotheses about the mean of a population. And today we'll be doing one sample t-tests primarily. So we'll be comparing what we collect from a sample, so the sample mean, we'll be comparing that to an established mean and seeing if there's a difference using statistics. So what I just said is using inference. It's when we use sample data to draw conclusions about a population. We can also use what we've previously been given about mu, the population mean, in order to make conclusions about a sample. So we go from a parameter to a statistic. So an example of this is if we were given the mean height for the entire UNC student body, we could use that to calculate the statistics of collecting a sample that has a certain mean. And we'll go through an example of that in the next slide. We can also use inference. So when we're given a sample, we can use that to make a conclusion about the population as a whole. So we go from a statistic to a parameter. And we'll be doing examples of both of those. So the first example is where we're using the mean of a population and using that to calculate statistics about a certain sample that's collected from the population. So we'll be in a particular hospital, the birth weight of newborns has a mean of 112 ounces and a standard deviation of 20.6 ounces. And we want to know the probability that a random sample of 10 babies has an average weight of greater than 126 ounces. So we know mu, the population mean, and we're trying to calculate a probability for the sample mean. So how we do that is we can use this z-test that you have learned about in previous lectures. And since we know that the population is normally distributed, so that's something we have to assume before we use this test, but since we've used the birth weight before, we know that it's normal. So here we have x bar, so that's the mean of the sample, which is 126, minus mu, which is the population mean, divided by the standard deviation over the square root of n. And we know that that's normally distributed. So since we know that, we can compute the probability using the R command. And here what this p-norm does is it gives the probability that with the normal distribution that it's less than that certain value, but we want it to be greater than the z-statistic since we want to look and see if the birth weight is greater than 126, so we do 1 minus p-norm. And that gives us a p-value, which we'll talk about later, of 0.02. So what that means is that there's approximately a 2% chance that we have, that given that the population mean is 112, that we would collect a sample that has a mean of 126. So it's not very likely. It's not very likely that the mean of 10 babies picked at random would be greater than 126 ounces. Now, an example of using the population mean, the sample mean, to make conclusions about the population mean is this next example that we'll be going through, which actually uses the exact same data that we had in the previous example. So here we're asking a different question, but we're using the same information. So suppose we believe that the average birth weight in the hospital is 112 ounces, but we aren't sure. So in other words, we wish to test the hypothesis that the population average is 112. If this hypothesis is true, what is the true probability that if we collected 10 babies, that they would have an average weight of 126 or greater. So here we collect the data, and then we want to see if it is different than what we would expect if our true mean was 112. So here we're making a conclusion about mu, the population mean, given a sample mean. So here we do the exact same thing that we did in the previous example, and here it's written out in a way that is usually how, when we test hypothesis, we'll have all of these written out. So first we have the null hypothesis, that the mean is 112. Then we have the alternative hypothesis, that the mean is greater than 112. We have our test statistic, which is that Z statistic, since it's from a normal population and we know the standard deviation, and that gives us a value of 2.15. And then we calculate the probability, which gives us the p-value. We're going to go through what the null hypothesis and the p-value, what those mean in greater detail in the next slides. This just gives you some terminology that's used in testing hypotheses. So the null hypothesis is that there's no difference between the population mean and the value that we're testing. It's often a straw man that we want to knock down. We want to refute our null hypothesis. So here are some examples of different null hypotheses that you can hypothesize, I guess. So in the first example, we have that mu is equal to 112. So here we have that mu is equal to a specific value. So for instance, if we wanted to hypothesize that, so here we have the mean birth weight is 112, or we could make any other hypothesis where mu is equal to a certain value. Then we can have mu equal to 0, so the mean weight gain from a treatment is 0. We can have mu t equal to mu c, so the mean from a treatment group is the mean from a control group. So these are just specific examples of different hypotheses you can test. And then the alternative hypothesis is that the null is false. So examples of this could be that mu is greater than 112, like we saw in the previous slide, or it could be that, for the second one, mu is less than 0, or it could be that the treatment mean is different than the mean for the control. And we'll go through an example of that not equal to, because that's called, it's a little different than what we did in the previous slide where we did the 1 minus pt. So we'll go through that in a little bit. And the p-value is the probability that, assuming the null hypothesis is true, that the test statistic would take values as extreme or more extreme than the value observed. So that's the probability that just by chance this would happen. So if you get a p-value of 0.1, it means that 1 in 10 times this could just happen by chance. Whereas if you have a p-value of 0.01, it's 1 in 100 times that this would happen by chance. So the smaller the p-value, the stronger the evidence against the null hypothesis. So these are the steps that we go through when we're testing a hypothesis. And we saw earlier how we did this in a specific example. But this is just a more general case that you can look at and apply to any sort of hypothesis testing that you're doing in your research. So first, you state the null hypothesis and the alternative hypothesis. And it's important to do this before you examine the data, because you might bias your results if you look at all the data and then you decide to make a hypothesis after that. So this is it's good to make the hypotheses first. Then you assume that the null hypothesis is true, and then we want to collect evidence such as finding the mean of the sample. and then we compute the test statistic and compute the p-value. And then from the p-value, we can see if there's evidence against the null hypothesis. So all of these steps, we want to refute the null hypothesis. So if we think that the null hypothesis is true, it's sort of, you can't really use these steps here. So this is one way to refute the null hypothesis, is using these steps. Now every time we test hypotheses, there's two errors that we encounter. The first type of error is rejecting the null hypothesis when the null is true. So just by chance, as we saw before with that p-value of 0.02, just by chance 2 times in 100 that it will happen when we have the mean being actually equal to 112. So that would be our type 1 error, is if the mean was actually equal, the mean for the babies was actually equal to 112, but for some reason the 10 babies we sampled actually had a much higher birth weight than 112. So it can happen. It happens a lot. You can so it's a type 1 error, but that's usually considered to be more serious. So we force the probability of type 1 error to be low, and that's the alpha value. Such as if you have a cutoff of 0.05 for your p-value, then 5 in 100 times you'll have a type 1 error. Type 2 error happens when we fail to reject the null hypothesis when the null hypothesis is false. So for example, if the birth weight actually of the entire population of babies actually isn't 112, but for some reason the babies we collect have a mean of around 112, then that will cause us to have a type 2 error. And when we conduct hypothesis testing, we try to minimize the probability of type 2 error, but type 1 error is the one that we control for the strongest. Now a quick talk about statistical significance. Like I've been saying, that the p-value is often alpha equals 0.05. This is kind of arbitrary. It's just a value that's decided beforehand, but in most papers you'll see that alpha equal to 0.05 is usually the cutoff point. So if the p-value is small or smaller than some value alpha, then we say that the data are statistically significant. So with our birth weight example, we have that p is equal to 0.016. So if we were using alpha at the level 0.05, we can say that it is statistically significant because it's less than 0.05. But if beforehand we decided that we wanted to use a cutoff point of alpha equals 0.01, then the value that we got from our sample with the p-value of 0.016 would not be statistically significant. Does that make sense? With the specific alpha you choose determines if the data you collect is statistically significant or not. So it's kind of with this alpha, it's kind of arbitrary. It's just something that's decided beforehand, but usually 0.05 is the cutoff point. So something we have to be careful about is deciding when something is significant or not significant. So if you have a p of 0.049, it would mean that we would reject the null hypothesis, but if we had a p of 0.051, it means that we would fail to reject it. So it's sort of, since those are very close together, it's sort of important to report p-values and confidence intervals so that the readers of your study know how close it was to being significant or not significant. So it's very, very important to report p-values, especially when they're that close to your cutoff point of alpha equals 0.05. So here's an example using the Urea data that we've talked about before. So suppose we wish to test the hypothesis that the mean urea excreted by cells is 30 milligrams per deciliter. So here our null hypothesis is that mu is equal to 30 milligrams per deciliter, and here our alternative hypothesis is that mu is not equal to 30. So this is a little different than before with the birth rate where we had greater than. So here we want to look and see if mu could be greater than 30 or less than 30. So we're looking at both sides of that bell curve. So I'll show you on the next slide. There'll be a little figure that makes it easier to see. So recall that if mu is equal to 30, then we can use this formula here where we have x bar minus mu over the standard deviation divided by the square root of n. And this is t distributed with n minus 1 degrees of freedom. And why we're using a t distribution is because we don't know the population variance or standard deviation. We just know the standard deviation from a sample. So this is almost always the case, is we almost always use a t distribution. So we use a t distribution when the data is approximately normally distributed and we don't know the standard deviation from the population. So here's that figure I was talking about. So here we will reject our null hypothesis if we get values that are in the red range. So the red range here is called the rejection region. So it's if we get values that are extreme to the right of our hypothesized mu, which is 30, or values that are extreme to the left of our hypothesized mean. And if we were using alpha equal to 0.05, which means that we're looking at occurrences that would happen 5% of the time, we would split that probability so that 2.5% of the curve is in the right red area and 2.5 is to the left in that left red area. And I'll show you how you can use R to do that. So this is using confidence intervals is basically what we're doing to see if we reject our null hypothesis. But don't worry, R will do this for us, so it won't be as tricky as computing it on your own and looking up things in a table. Because I know if you've taken stats class before, you've dealt with trying to find the table and go through and find your degrees of freedom and all of that, but R makes it easy for us because we have other things to do with our time. So this is one way you can use R to calculate this for you, and this is a little more complicated, so don't worry about memorizing this right now, but this is shows you step by step basically how to do it. So first we use that t statistic that I talked about before, where you have the sample mean minus the population mean, which is 30. That's what the null hypothesis is, that mu equals 30, divided by the standard deviation from the sample, sorry, and the square root of n, which is how many data points we've collected. And here the PT gives us the probability that it's less than that value on the curve that we saw before. And we have to specify the degrees of freedom, which is n minus 1, so it would be 26. And here we do 1 minus PT because we want the area to the right of that. So we want the probability that it is more extreme than the value for the t value that we've collected. And we do 2 times this value since we're looking at both of those extreme values on the curve. So the extreme values to the left and the extreme values to the right. And this gives us a p-value of 9.7 times 10 to the 6th, so a very, very small p-value. So this is, by showing this to you, it gives you a way to actually see how using the confidence interval things that we have talked about before, how it will give us this p-value. but we can use the t-test command to save us some time, and it will do the exact same thing for us. So here we use t-test and then the data that we're looking at. So in this case, the column would be labeled urea, so it would have all of the data points under that column, and then we specify what mu we're testing. So we specify mu equals to 30. And then if we look at this t-test, so first it gives us the t-value that it calculated. So that's the x bar minus mu over the square root of the standard deviation divided by n squared, the square root of n. And then it calculates the degrees of freedom. So since we have 27 points, the degrees of freedom would be 26. And then it gives us the exact same p-value that we saw on the slide before. So here, this does the test for us, so it even says that the alternative hypothesis is that the true mean is not equal to 30, and it gives us the 95% confidence interval as well, and the mean of x. So it gives us all the data that we want to look at, all the statistics that we'd like to look at from this data set for testing this hypothesis. So some quick notes about what the t-test function does. So here x is the data set that we want to look at. Mu equal to mu 0 is the specific population mean that we're testing. So if we omit the mu in this function, it'll automatically test that mu equals to 0. But if we're just using the t-test function to calculate a confidence interval, we don't need to worry about specifying mu. So if you're just wanting to use this test to save some time and calculate a confidence interval, you don't need to worry about specifying a mu value. So what we just did before was a two-sided hypothesis test. So that's when we're looking at the critical regions for the extreme left and extreme right of our hypothesized mu. But we can also do one-sided hypothesis tests. So for instance, here we could, our null hypothesis could be that the mean concentration of urea is less than or equal to 32 milligrams per deciliter. And then our alternative hypothesis is that mu is greater than 32. And this is a one-sided hypothesis test. So when you're making a null hypothesis, you always want mu to be equal to or less than or equal to or greater than. So mu, your null hypothesis always has some sort of equal in it, and your alternative is not equal to or greater or less than. So just something to clarify there. So if we're doing a one-sided hypothesis test that mu is that our population mean is greater than a specified value, we want to look at the rejection region to the far right of this bell curve here. And if in this case, if we had alpha equals to 0.05, then this red region would illustrate 5% of that entire bell curve. So if we get values within the 5% extreme right of this bell curve, then we'll reject our null hypothesis. Does that make sense? Any clarification needed there? Okay, I'll keep going. Because I know this can be kind of tricky with the rejection region, because I've struggled with that too when I first learned statistics. So if we wanted to do a one-sided t-test, we can also have R do this for us. So we saw before how we can use PT, the PT function, to calculate a p-value. So that's putting in the entire the t-test, calculating a t statistic, and then looking at the probability that it is greater than that statistic. So here you have the mean from the sample divided by the hypothesized mu over the standard deviation of the data that we collected divided by the square root of n, and then we specify the degrees of freedom. And here we don't do a 2 times this value because we're just looking at a one-sided. So we don't need to do that. And we see that that gives us a p-value of 0.001. Now if we use the t-test function in R and we specify alternative equals to greater, then that gives us the exact same p-value that we saw above when we basically calculated it by hand using R. So this t-test function has a lot of functionality, so we can use it for any type of test when we're using a t-statistic. So when we assume that data is normally distributed and we don't know the population standard deviation. So this is a quick reference slide that you can look at when you're working with data, like as in the homework, figuring out how to specify if you're testing using one-sided tests or a two-sided. So if you want to hypothesize that your alternative hypothesis is that mu is actually greater than your specified value, then you just plug in alternative equals to greater. If you want to look at that it's less than the value, you specify that there. Or if you want to do a two-sided test, then you can also enter that into the function. And these are all for your specific mu 0. Okay. So why we use two-sided tests is that it's a lot more conservative than a one-sided test, and almost all of the time you'll be using a two-sided test. You see that a lot of times in research. In studies that you've read, they state the p-value and then a confidence interval, which is used generated from a two-sided test. But you can use a one-sided test if your sample is small, but you have to be able to defend that decision. So it's important to make the decision to use a one-sided t-test in advance, because if you get your value, your t-value afterwards, and then you decide, oh, I'm going to use a one-sided test, it may give you significant results, whereas if you were using a two-sided test, it might not be significant. So it's something you want to consider beforehand, before you collect the data, before you run the tests. So as we talked about before, when you have alpha equals to 0.05, it means that it's significant only if mu 0 is outside the 95% confidence interval for mu. So when we saw before with that graphic with the two red regions for the extreme right and extreme left for mu, it's only significant if it's outside of that confidence interval, so if it's in those red regions. But if we get a mu 0, so that's the hypothesized mean, if that's within that confidence interval, then we can't conclude that, that we can't reject that null hypothesis. So to make this more clear, I'll use an example here using the Urea data. So in the first t-test here, we test that mu is equal to 33, and we don't specify, we're not specifying a confidence level, so it gives us the 95% confidence interval. So if you don't specify what confidence level you want to look at, it'll automatically do 95%. So that gives us this confidence interval of 33.2 to 37. And since our hypothesized value of mu is 33, it's not in that confidence interval, so we would reject the null hypothesis. And you can see this p-value is 0.03, so it's less than the cutoff of 0.05, which is what the 95% confidence interval means. So if we have, in this next example, we have a confidence level of 0.99, and that would mean that our alpha would be 0.01. So alpha is whatever confidence level you want to look at. Alpha is 1 minus that. So if we had a 90% confidence interval, our alpha would be 0.1. So is that clear? Different alpha and confidence levels. Usually in when you're testing, they'll usually specify what confidence level you want to look at, so you can just put it in this t-test function here. So for this second part, we see that we specified that the confidence level is 0.99, and then the 99% confidence interval that it gives us is 32.5 to 37.7. So our mu, our hypothesized mu, is actually in that confidence interval because it's 33, so it's right in the middle. So it wouldn't be significant. And we can, since our alpha is 0.01, 1 minus 0.99, 0.01, and the p-value is 0.03, we can also see that it's not significant there. Okay, so is that clear? So it can be a little tricky first learning this, but hopefully when you work through data, you'll get a hang of it and see how you can use R to save some time and it gives you all the information you need here too, so you can analyze that. So when we say something is statistically significant, it means that the result is likely not due to chance. So it depends very heavily on the number of observations that we collect, because as we've seen for that z-test and the t-test, we divide by the square root of n. So it's important that we have large sample sizes if we want to be able to have statistical significance in a test. But practical significance is a little different than statistical significance, because the results you gather might be very extreme, but given that you only had 6 people, it might not show up as statistically significant. So an example of this is in cholesterol drugs. So if we had a study with 5,000 patients, and drug 1 lowered LDL cholesterol by 1 point, and then we had another study where we had 50 patients and drug 2 lowered LDL cholesterol by 25 points, we can see that this second study, the drug seems to be a lot more effective than the drug 1, but quite possibly drug 1 is statistically significantly better than placebo, and drug 2 might not be, and that just has to do with how many people were in the study. So here again, it's important to report p-values and significance, so readers of the study can understand how close it was to being statistically significant, or what these, or how many points the drug lowered LDL. So it's another thing to consider when you're running doing hypothesis testing. Is about sample size and how that might make something statistically insignificant, whereas it really does have an important meaning. So power is another consideration in hypothesis testing. So the power is defined as the probability to reject the null hypothesis given that the null hypothesis is false. So what type of, here's a quick little pop quiz, what type of error is it if we fail to reject the null hypothesis when the null hypothesis is false? Yep, type 2. Good job. So the greater the power, the less chance of a type 2 error. So when we have high power, it implies that we are likely to detect a truly significant result. But if we have too small of a sample, our test might have insufficient power, so we won't actually reject the null hypothesis when it's actually false. So oftentimes when you're proposing, writing a grant proposal, funding agencies ask you to demonstrate that the proposed study has a sufficient power to test your hypothesis, and that's something you can do with simulations, things like that, but we're not really going to discuss how to use simulation to do that, but it's something fun to look at if you're really interested in power calculations. So as we talked about before, in most circumstances we reject the null hypothesis if p is less than 0.05. But if we're trying to overturn a widely accepted hypothesis, a more stringent threshold may be necessary. And in general, it's dangerous to publish an unexpected result simply because the p-value is 0.05. So this has to do with what you're testing, basically. So if it's something such as showing that some like vitamin C is beneficial for health, it's something that won't cause any harm if people start taking vitamin C. So if you have a p-value is 0.049, you might publish it because it's statistically significant that alpha equals 0.05. But for instance, if you have a drug that could cause a lot of harm, a lot of side effects, and you want to show that it's better than placebo at like a cholesterol drug, but it causes a lot of side effects, and you have a p-value of 0.049, it might not mean as much as the vitamin C. So something to consider when you're testing hypotheses, just based off of what you're trying to show. So it's important, another reason why it's important to show p-values. And a problem that we can have is when we test multiple hypotheses is that if we reject the null hypothesis, when p equals 0.05 for each test, and we're wrong 5% of the time, that this can cause us to publish or report data testing that actually isn't significant. So it's a major problem when we're testing multiple hypotheses. So an example of that is this schizophrenia example we have here. So if researchers gave psychological questionnaire with 100 questions to schizophrenia patients and matched controls, and so if 4 of those questions, the responses were significantly different between cases and controls at p equals 0.05 level, and then they immediately publish their results. So can you see what the problem here might be? Anybody want to try to explain it? No. Okay.
For questions, it's 4% questions.
Okay.
They didn't do multiple testing.
That's true. Yep. So for each question, 5% of the time, you would give a result that's different, that would be more extreme than the values. So that's your type 1 error, is when you reject the null hypothesis when it's true. And so that happens if you're using a p-value for each question of 0.05, that happens 5% of the time. But if you're looking at 4 questions, it would happen that for each of those questions, the value you get would be more extreme, much more. So you'd want to make sure for each test that if you add them all, if you look at them all together, that 5% of the time you get values more extreme. So it's a little confusing, but we'll talk about it more in in future lectures. But so just the important thing to remember here is if you're testing multiple things, you want to have a more stringent p-value than 0.05. So like it said, when testing multiple hypotheses, the probability of making at least one type 1 error is much greater than 0.05. So this is a major problem in modern bioscience, when we're testing a lot of hypotheses about genetics at a time. So in high throughput genetics such as microarrays, GWAS, etc., this is a big problem, and that's genome-wide association studies. So we'll talk about this in the future, but for now, if you're performing multiple tests, the p less than 0.05 threshold might be too low. Another word of caution is the results of hypothesis tests are only valid if your sample is representative of the whole population. So that goes into biases. So if you only select people that are extremely tall in your sample, when you're testing the mean of a height of a population, that could be a problem. You want to make sure that each observation is independent of others. And for small n, the data must be approximately normally distributed, to use the t-test that we talked about before, and it's important to watch out for outliers. So here's an example of what can happen when we have outliers in our data. So this is that Urea data, and suppose instead of typing in 31.29, we accidentally type in 3,129. And then we want to test the hypothesis that mu is equal to 30, so we use this t-test. So here we get the mean of our sample is equal to 149. And it gives us this huge confidence interval from -85 to 385. And we get a p-value of 0.3. So it means that we don't get significant findings. So we can't reject the null hypothesis that the mean is equal to 30. So that's a big problem when we have outliers. They can cause us to conclude things that aren't true. So even a handful of spurious observations can reduce the power of our test, or even cause us to falsely reject the null hypothesis. So it's important to check for outliers before performing tests, and it's another reason to report confidence intervals along with p-values, because if you have a very wide confidence interval, it's often a warning sign that there's outliers in your data. So some points to remember from the lecture we're going to go through now. So are there any questions, anything that needs clarification before we do a quick wrap-up of what we learned? Okay, so we've seen today that one sample t-test can be used to test hypothesis that the mean of a data set is equal to a certain value. So that's all we're doing today is one sample t-test. So by convention, we call a test significant if p is less than 0.05. But we need to be careful about that because it really has to do with the type of what we're testing, what the data is collected about, things like that. And there's a close relationship between confidence intervals and hypothesis tests. And it's advisable to report confidence intervals along with test results and p-values. So when we test hypotheses, we're looking at those confidence intervals that we've learned how to calculate before, and that's how we make conclusions. Just because a test is statistically significant doesn't mean that it's practically important. And also, the opposite is true, too, that it might be practically important, but just since our sample size was so small, we couldn't get statistical significance. So that's something else that we need to consider when we're doing hypothesis testing. And then when we're performing multiple hypothesis tests, we need to be extra careful about making conclusions. Then here's some cheat sheet of our commands to remember from the lecture. So here are the different t-test specifications. So first, this t-test performs a two-sided test of the hypothesis where the mean is specified here, so mu equal whatever value is the test that you're performing. And if you don't specify the alternative, it will do a two-sided. But if you want to do greater, you can specify that, or if you want to do less than, you can specify that as well.